Hello, welcome to Biotanemy.
DNA isolation. DNA isolation or DNA extraction is a process of purifying the DNA using physical or chemical methods from the sample. There are 5 basic steps for DNA isolation. 1. Disruption of the cellular structure. That is to release the DNA or RNA from the nucleus. This can be done by physical, chemical, enzymatic, or by combination of the 3 methods. The physical method involves sample grinding or crushing to disrupt the cell walls or tough tissues present in the cell. The use of heat can also help to disrupt the cell membrane. The use of chemicals such as detergents, cheotropes will also help to disrupt the cell membrane. The enzymes like proteinase K, collagenase, zymolase, lidocase, lipase, and lysozyme. The second step of DNA isolation is to clear the lysate or to remove the cell debris, separating DNA out of them. This can be achieved with the help of centrifugation and chemicals like phenol-chloroform. The cell debris and proteins can be precipitated out using a high concentration of salt solution. The precipitated cell debris and protein can be separated out by centrifugation. The DNA will be precipitated out by adding isopropanol. This will precipitate large DNA molecules, while RNA fragments, which are smaller than DNA fragments, will remain soluble in the solution. The pelleted DNA fragments are then separated via centrifugation. The pellet is then washed using ethanol to remove salt and to enhance evaporation. Finally, the DNA pellet is again resuspended in an aqueous buffer such as trichdTA or nuclease-free water. The procedure for DNA isolation. Step 1. Transfer 2 milliliters of overnight bacterial culture to a sterile Eppendorf tube. Harvest the cells by centrifuging them at 12,000 rpm for 10 minutes. Step 2. Collect the cell pellets by gently discarding the supernatant solution. Onto the pellets add 600 microliters of lysis buffer, which consists of 440 microliter TE buffer with a pH of 8 and 45 microliters of 10% SDS solution and proteinase K. Gently invert the tubes to mix the solution. And incubate them at 37 degrees Celsius for 1 hour. Step 3. Add 500 microliter of phenol-chloroform mixture. And mix until the solution turns into milky white. Centrifuge the solution at 10,000 rpm for 10 minutes at a temperature of 4 degrees Celsius. Step 4. The supernatant is collected this time and the above-mentioned step is repeated. Step 5. Add 5 microliter of sodium acetate of pH 5.2 to the collected supernatant. Mix the solution gently and thoroughly. Step 6. To the solution add 1 milliliter isopropanol solution. Centrifuge it at 1000 rpm for 5 minutes. Step 7. Collect the DNA pellets by carefully inverting the tube. Air dry the tube. Finally, dissolve the DNA pellets in 50 microliter TE buffer in a pH of 8.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.